I'm excited to be joined today by Dr. Emily Jacobs. She is an associate professor of psychological and brain sciences at the University of California, Santa Barbara, where she studies how sex hormones influence the brain, particularly during the menstrual cycle and during menopause. Emily, welcome to the Nature and Nurture podcast.
Thank you so much for having me. Thrilled
Was
to be here.
that an accurate description of your research? Is it primarily in females only, or are you looking at hormones in males as well?
Yeah, no, we do males as well. We study the effects of endocrine aging on the human brain, and that includes males and females. So, one of the biggest initiatives right now in my lab is um, thinking about the midlife period and the changes in the brain that happen really sort of early in the traditional aging process. And we have all genders and
So,
sexes.
so menopause is something that most people have heard of. You mentioned off camera andropause, which I guess is an androgen-based male equivalent. I've never heard of that before. Could you explain?
Yeah, so endocrine aging is a phenomenon that happens in males and females, and in humans, the decline. So endocrine aging meaning the decline in the production or secretion of sex hormones with age. In men, it's very protracted. So men will start to lose testosterone production beginning at about age 30. And there's this sort of slow, steady decline that doesn't stop until death, right? Men are still secreting testosterone forever, essentially. But you do see this sort of protracted decline in production. So that refers to andropause. Menopause is something special in the sense that it's also an overall decline in the production of sex steroid hormones, but it happens over a much more narrow time window. And so we can start to disambiguate the effects of chronological aging from endocrine aging much easier in females than in males. Now, there's all sorts of other ways we could start looking at the role of hormones in the brain, doing more direct interventional studies, but just looking at spontaneous decline, it's hard in men to figure out what might be driven by the decline in hormones versus just the ticking of the clock, just chronological age. But yeah, andropause is definitely a real phenomenon.
And you're at UCSB, and there are a lot of evolutionary-minded thinkers there. Does that come into your research at all, thinking about like making predictions based on evolutionary theory or cross-species comparison based on at what rates or at what ages you would expect these hormone changes to happen?
Adam, that is a great question. And really, I look forward to having much more conversations. Lita Cosmides, who of course is sort of a pillar in this field, and I have had really fun cocktail discussions, but we haven't yet put our heads together. We have brainstormed some ideas, but we have not yet put our heads together in a formal sense. So I would be really excited to go in that direction. I'm more in the camp of I kind of grew out of traditional cognitive neuroscientists in that pool, and kind of pulling in neuroendo into that piece. But yeah, there's a million directions we could go, and it's so fun to be at a place like UCSB that has just different lenses for thinking about phenomenon. But no, that's one that we have not explored yet.
So the first line of your research bio is the brain is an endocrine organ, which is absolutely true. But in I didn't really learn that until I started doing hormones research myself. Like in traditional cognitive neuroscience, I guess more of the focus is just on these other non-hormone based changes, whether that's like genetic or experience driven, neuroplasticity type stuff, much less focus on the brain as an endocrine organ.
Hmm. Yeah, I mean, I when I joined Mark Desposito's lab at Berkeley as a PhD student, when I was in your shoes, right, my first year, um, his lab was really interested in dopamine, right, and how this major neuromodulator influences uh, cortical tissue and cognitive functions that are dependent on the prefrontal cortex. And this is a little bit of a backstory, but I will get around to answering your question. I came from Smith and had great neuroscience training, but didn't know a lick about dopamine or really neurochemistry in general. I had not taken any kind of super in-depth course. So I joined Mark's lab, and my first act for the first 6 months was basically to lock myself in an office and just pull any paper that had the word dopamine in the title. And, whether it was like gene knockout studies or patient studies of Parkinson's, patients on off medications, like whatever, I didn't care. I just wanted to immerse myself in this literature. And it was during that period of time that I stumbled across this beautiful body of work by Jill Becker at University of Michigan. And this was work emanating from the 90s. And she had published just a series of beautiful studies, both in vitro and vivo. But the central takeaway was that endogenous fluctuations in the sex steroid hormone estradiol being the major form of estrogen in most mammals, that these endogenous fluctuations can provoke or stimulate or potentiate dopamine release in the mammalian brain. She was working in rodents. And I was like floored, because that, having just spent 6 months reading everything about the dopamine literature heavily geared towards the human, I had never once come across the fact that hormones might tell us something about the organizational principles of the brain, or certainly this interaction between our endocrine system and the traditional neuromodulatory systems like dopamine. And that just opened me up into this whole world of endocrinology, and specifically neuroendo, the formal study of which was really kind of launched in the 1960s. But we've known something basic about the endocrine system all the way back to the time of antiquity, right? We've used castration as a form of changing animals' behaviors. But really, what I saw was this major conversational gap between what I'll say the sort of traditional cognitive neuroscientist and those scholars who are working in the endocrinology space. And I think there was this amazing kind of merger of those two fields, at least at the animal level, in the 1990s, not just from Jill's work, but also this woman, Catherine Woolley, who was I think she was, no, she's not at Columbia. Where was she? Johns Hopkins, I think, at the time. And she published one of my favorite research papers of all time. She was looking again in rodents and showing changes in hippocampal spine morphology. So these are CA1 pyramidal neurons, and you could see changes in dendritic spines based on estrous cycle stage. These are cross-sectional studies, but what she shows is that during proestrus, which is the stage of the rodent estrous cycle when estrogen levels peak, that's coincident with this proliferation of spines. That was a turning point in the field, because my understanding is that before that point, a lot of endocrinology was really focused on hormones' roles in overtly reproductive behaviors, right? Like lordosis behavior in rats, for example. And she really started to make this case that no, sex hormones are powerful neuromodulators that have effects in what we call extra hypothalamic sites, right? Not just the hypothalamus, but in hippocampus, and later discovered in prefrontal cortex. So these are modulating cognitive functions. That happened way back in the 90s, and we can get into that literature more if you want. But the human literature didn't really catch up for another 20 to 30 years. But we're now filling that gap.
So neurotransmitters and hormones are both really just chemicals in the body, right? And it seems like it's more of a context-based thing. Like we call something a neurotransmitter if it's primarily active in the brain, and we call something a hormone if it's primarily active in the blood or other areas of the body. Is there any true categorical difference other than like where you find it? Because some chemicals are both neurotransmitters and hormones, right?
Mm-hmm. Yeah, there's neurohormones, right? Neurosteroids. So the brain has all the molecular machinery it needs to produce hormones. So there's a couple ways to think about the difference. One is just the distance in which these different chemicals act. So with traditional neurotransmitters or neuromodulators, it's they might get packaged in vesicles and diffuse exocytosed into the synaptic space, and then they'll diffuse across like a 20 to 30 nanometer synaptic cleft, and then they bind to receptors that are like right there, kind of primed to receive it. Hormones are acting over much larger distances, right? Like 1 to 2 meters. They use their circulatory system as the superhighway, so they're going everywhere your blood goes. And the magic becomes the distribution pattern of where those hormone receptors are expressed. But but they are also, some hormones can be packaged in vesicles just like a neurotransmitter. Others, like steroid hormones, can't because they're lipophilic. But I would say the major distinction is really just thinking about the specificity and the sort of duration in which, not duration, but the distance in which these chemicals are able to travel.
And what about
And
pheromones? Is that the same thing but transmitted through air?
yeah, okay, so here's the thing. We are multicellular organisms, right? And any multicellular organism, whether we're talking about like a sea slug all the way up, multicellular organisms have to have a way for their cells to communicate. And as you just mentioned, like neurotransmission is one form. You can have hormonal forms of communication. And then in some instances, right, you can actually have these instances where there's secretion of chemicals outside of the body, so that there's sort of like organism to organism communication. And this is it's a cool concept. And I don't know that literature too much, but it's certainly wild to think about, like maybe like where I end and you begin is more blurred than we really think.
So going back to your PhD, you mentioned you're at Berkeley, you were reading everything there was to read about dopamine, and then you had this epiphany realizing how hormones are interacting with typical neuromodulators. And then your research from there transitioned in more of a neuroendocrine direction.
Yeah, so I stumbled on this beautiful work of Jill Becker's, linking this modulation of estradiol with this ability to potentiate dopamine release. And I was totally taken because, as I mentioned, there's just such little research from the human cognitive domain thinking about the role of hormones as these powerful neuromodulators of learning and memory. There's also work at the time by Rebecca Shansky and Amy Arnston thinking about this notion of like the inverted U, so dopamine, especially when it comes to thinking about like working memory, that there's sort of this sweet spot, and too little dopamine or too much dopamine is associated with sort of poor working memory, but there's this kind of sweet spot. And they had shown some really elegant studies in which modulation of estradiol could kind of move the animal's performance based on sort of how much estradiol induced dopamine there was. I won't get into the weeds except to say that I basically took that preclinical finding and just translated it into humans to say, like, can we see the same thing across taxa? And sure enough, we did. I mean, it was it was in retrospect a very risky thesis, but we found this evidence of estradiol modulation of dopamine dependent cognitive functions, and that really convinced me that this is probably a real phenomenon, that sure enough, these steroid hormones deserve much more attention than they have traditionally been given in cogneuro. And so I just continued to kind of chase that thread.
When you say modulation, does this mean that you're looking at natural endogenous hormone levels and then just somehow able to trace the function to the brain? Or are you doing something more like a control trial where you have a placebo group and a drug group, and in the drug group you administer hormones and then you look for differences between groups?
Yeah, so we can gather we can build this evidence from a lot of different methodological techniques. So that early study was looking at just endogenous changes within women. So this was a study that followed women at two different points in their cycle under low estradiol and high estradiol conditions, and seeing sort of differences in prefrontal cortical function. And there was another twist in there that had to do with genetics that I won't get into. In the studies that we now run, we do pharmacological manipulation. So here's a little neuroendo for I guess the listeners. Your brain controls the production of gonadal hormones, and it does so through this hypothalamic pituitary gonadal, or HPG axis. So a lot of people have heard of like the HPA stress axis. Well, there's a parallel axis for regulating the production of gonadal hormones, so testes in men and ovaries in women. And so the hypothalamus has about 1,500 to 2,000 neurons that really guide this axis. These are gonadotropin releasing hormones, or GnRH neurons. And what's so cool is that, I don't know, maybe we'll get into the neurobiology of puberty and like how these neurons like come out of quiescent, but in reproductively aged animals, these neurons fire these classic kind of like pulsatile release to stimulate that whole axis. So knowing that, we can use drugs, we do use drugs that act on those 1,500 to 2,000 neurons to shut down the entire axis. These are gonadotropin releasing hormone agents. And we're running a study right now following women before drug use and then about 3 to 6 months into use of this drug. And it is akin to, if I can be a little bombastic, but it's akin to chemical castration. So women who go on this drug, these are often 20, 30, 40-year-old women, will experience menopausal symptoms within a matter of days. And they have sex steroid hormone levels that are indistinguishable from that of a postmenopausal woman. So it is a form of pharmacologically induced menopause. And what we're doing is looking at the brain's response to that new endocrine condition. How does brain structure, function, cognition, how does this all change in response to? So that's one example. There's a lot of other ways we can think about tracking the relationship between endogenous modulation in hormones and the brain. And one of the most exciting studies that we wrapped was this project called 28andMe, and obviously a riff off of 23andMe.
Mm.
But one of the, and maybe you're sort of alluding to this, but you know, kind of one of my quarrels with traditional cognitive neuroscience research is that often you'll take, and this isn't just for endo-based studies, like anything that you're interested in, like you'll take a group of people in one condition and maybe you'll take a group of people in another condition and you average their brains. Like let's say a classic aging study, right? Like you'll take young adults and older adults and you're kind of like average their brain and make some inference about the difference. That thinking about how variation in hormones might affect variation in some aspect of the brain, because a central feature of the mammalian endocrine system is that hormone secretion varies over time, right? There's these sort of classic ebbs
Mhm.
and flows of hormone production. And no doubt it's the delta that matters a lot more than the relative concentration. So if you just take like a canonical menstrual cycle, you're going to see an 8-fold increase in estradiol during around the ovulatory period. You're going to see an 80-fold increase in progesterone throughout much of the luteal phase. And those pulses, right? We like to say that those pulses are almost like a vital sign. Like these are rhythms that drive physiological functions. And yet we had almost no insight into how those rhythms might drive change, or how the brain might respond to those changes, in part because we were sort of plucking people right at these like arbitrary points or these sort of singular points in the cycle, as opposed to really capturing the entire rhythm.
Mhm.
And that, so we started sort of, you know, this is a project led by a brilliant graduate student, Laura Pritchett, and a whole bunch of other amazing minds. We started kind of like chewing on this problem a little bit in the, you know, at lab meetings or sort of in the background. And then we sort of watched as a lot of groups, a lot of traditional cognitive neuroscience groups, you started to see this flip over the last 5 to 10 years, which is this, maybe 5 years, like this adoption of these highly dense sampling brain imaging studies. So this really sort of flipping the model of like, instead of taking 30 people, scanning them once and averaging their brain, you take one person and you know, image them in just incredible with incredible precision. So this was really led by Russ Poldrak's My Connectome project, but also, you know, the Midnight Scan Club. So it's this idea of precision imaging of small groups of people, but with, you know, over really unprecedented timescales. And that was when we had this epiphany of, oh, this is the perfect approach for you know, our problem, which is trying to understand these rhythms of the brain. And so 28andMe was designed to do just that, to understand how the modulation of hormones might drive or impact aspects of the brain.
Emily, do you know Katie Bottenhorn?
Yes.
So she's
Uh,
a postdoc in my old lab at USC with Megan Herting. We talked about this dense sampling approach. She was doing, I imagine, a similar study to what you're finding in 28andMe. So, and the part that blew my mind is that across the cycle, you're not just seeing changes in brain function or connectivity, but even structure is changing. And I tend to think as brain structure is this fixed thing.
Yeah. Yeah, right. I mean, we're all conditioned to think that. In fact, if you crack open a textbook from even 20 years ago, it'll say, like the human brain is basically done by age 2, right? And then there's a whole wave of adolescent, cognitive
Yeah, I should
development.
add so my advisor doesn't get upset at me because we study adolescent brain development. I mean,
Okay.
once I think of structurally an adult's brain, then
No,
I
no,
think
no.
of it as fixed.
No, 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 but for sure, you were ahead of the curve in this sense. But most of the field 20 years ago, 30 years ago, used to think that like the brain was relatively fixed. And then, folks like your former advisor, I think of like, Jay Geeds, longitudinal study, or Sarah Jane Blakemore, like these folks and many, many others really can, showed that adolescence, right? This period of time that's coincident with hormones coming online is a period of incredible synaptic plasticity in the brain. Okay, so that was fixed. And by the way, like anybody who's parented a child through puberty kind of knows that the brain must be changing. But then after that, it was kind of like, okay, well, get adolescence out of the way, and then it's really dumb. The brain is fixed, it's not changing. Couldn't be further from the truth, right? The brain is like highly dynamic, and that's what I love about these dense sampling studies, is that we can see these changes over almost unprecedented timescales. So my postdoc Caitlin Taylor published a paper in Neuroimage a couple of years ago, taking this approach and looking at, so using high resolution imaging of the hippocampus to look at hippocampal structure across the endogenous cycle, and then in response to pharmacological manipulation, where she selectively suppresses progesterone by 97%. And sure enough, like the modulation that you see across an endogenous menstrual cycle is completely eradicated when you remove that hormone cycling. And we've kind of just moved into this space of thinking about pregnancy too. So there's beautiful work from groups in Europe. So Susanna Carmona, Elceline Hexama, Magdalena Martinez, showing that pregnancy is again this period of just incredible and dynamic plasticity, both in structure and in function. And we're applying some of those precision imaging lens right now to track how the brain responds to these just, amazing, changes. So if you think about the hormone changes that occur across the cycle, that is but a blip on the radar of the magnitudes of changes that happen across the gestational window. And sure enough, the brain is changing. These hormones are guiding, I think, right? What we're seeing is that the hormones are guiding the plasticity and structural remodeling of the human brain over this time course, and it's doing so with good reason, right? You mentioned sort of what are the evolutionary functions of these hormonal signals. Well, here's the most obvious one. When estrogen and progesterone levels surge across the 40-week period and then drop precipitously at delivery, we can track changes in default mode network, so really sort of across the brain. And no doubt these changes are part of what are guiding these behavioral changes that help the new parent parent for the first time. If you've
It's
ever had a dog who's given birth, um, you've witnessed this, right? So you might have had like the most docile, kind creature. And then all of a sudden, if she gets pregnant, like a couple days before birth, she'll start to dig a nest, right? This could be an unusual behavior for her. She'll start to dig a nest, she'll deliver. And then if there's some kind of intruder, whether it's an animal she's seen before or somebody else, like she can display signs of maternal aggression, right? For a previously completely docile animal. This is the role of hormones, right? Like it's really helping the animal care, give, and respond to this new situation.
it's really interesting seeing those behavioral findings dovetail with the neurobiology that you're describing, because I was completely exposed to endocrinology research from psychology at first. So, the classic findings of like testosterone being linked to aggression, or oxytocin, the love hormone, and all this stuff, but definitely missing the piece of the neurobiological why, or what is actually happening here.
Yeah.
Now, in your own
That's
research, when you're finding these structural or functional brain changes, is it always associated with some cognitive task, or is it like mapping out the basic what and then maybe why questions to be asked later?
Yeah, if we kind of divvy this up into the ultimate versus proximate questions, right? Ultimate being the like why and the proximate being the how. My lab is definitely more deeply, our center of gravity is more into the how to then help us move into the why. Um, so we do there are situations in which we are really interested in potential cognitive changes. For example, across the menopausal window, we can see pretty consistent changes in aspects of verbal episodic memory. So things like a list learning task or face name associative memory tasks. And across different cohorts and pretty diverse cohorts, like that's a pretty consistent finding, which is that these abilities show a modest but statistically significant and consistent decline from in postmenopause relative to pre and perimenopause. Um, which is to say that's a pretty specific effect. When we think about endocrine aging, there's other domains like working memory that don't show consistent changes or selective attention, but probably episodic memory or even aspects of spatial navigation um, appear to be changing over this period of time. So yeah, we explore those behavioral endpoints at times and then ask what are the neurobiological mechanisms that lead to those. And other times we're just interested in like how are hormones coming in and sculpting the brain, and then can that give us insight into what the adaptive functions of those changes might be.
There are these why questions for the neurobiological how that you're describing, and then there are sort of meta why questions. You had a TED talk where you talk about um, kind of the
Mm.
history of neuroendocrine research and sex and gender bias that happens there. Like historically, most research, especially animal research, was only focused on males. And then even when focused on females, maybe it's more centered on puberty and menstruation starting, but less so later in life and focusing on menopause and these age-related changes in endocrine function over the long term. Could we introduce some of the history there, fill in those gaps?
Yeah, yeah, sure. So this is, literally my soapbox point, I guess you could say at this point, which is just this interesting question of why is it that women's health is arguably 20 years behind where it should be? And I think this is where you have to think about kind of the intersection between, the status of women in STEM and the status of women's health research. And I think those are concurrent phenomenon, as well as connected ones. Those are not just existing in parallel; they're really kind of informing one another. And what I mean by that is that, if you sample current tenured professors in neuroscience, you'll find that about 80% of them are men. Um, just to stave off any criticism, I'm not suggesting there should be less men. I'm just suggesting, as this conversation continues, that maybe there's a real argument that there should be more women. Right? Um, I think that lack of representation impacts the nature of the questions that we ask. For example, um, there is a sort of now classic finding from a group in Berkeley. This is Annalise Beery, Irv Zucker, who kind of started talking to colleagues and noticed that, hey, is everybody else relying on male animals in preclinical research? Because we're kind of noticing this local pattern. And sure enough, so Annalise led this survey where she goes into 10 different fields and takes, some number of the top journals in those fields and just flips through the methods section and asks what was the sex of the animal used in that research, looking back historically. And overwhelmingly, she observes that this preclinical research, so in animals, is relying almost exclusively on male animals. And the biggest offenders were neuroscience, immunology, and pharmacology, right? Arguably, like three fields that could have a tremendous impact in human health. So there's this. So she kind of quantified the sex bias in basic science, And you might add, I think this is a phenomenon that the public probably is not aware of, but it is true, and it's been true for 50 or more years. And here's the rub, that sex bias in basic science is impacting human health, and it's probably doing so in ways we don't even fully understand. Um, so, for example, we know that most of the drugs that are pulled from the market by the FDA are done so because they have adverse effects in women. Presumably, these are effects that would have been, discovered had that early preclinical testing included both sexes. So that's one example of, I think these biases that can happen. And I think it's interesting that, here was this woman, Annalise, who noticed this issue, and it got splattered across major news headlines. There was Nature and Science editorials, and it led to this kind of renaissance. So Janine Clayton, as the director of the Office of Research on Women's Health, really pushed hard for this SAB, sex as a biological variable, policy change, setting the new standards for any NIH proposal. And I think it's not lost on me, and hopefully it's not lost on the audience, that here were two women in positions of power that noticed this bias and did something about it. And this bias had been going on for 50 years, right? And we can look at other examples. So in my field of cognitive neuroscience, we are talking about a little bit earlier that one of the major questions is trying to understand how the brain changes with age. And like writ large, the vast majority of studies are focused on this model of thinking about enrolling men and women who are 65 years and older and comparing aspects of their like brain chemistry or brain anatomy or brain physiology to young adults, usually psych undergraduates, which is a whole other problem, probably. So, but like, that model of thinking about young versus, 65 plus has told us a lot about how the brain changes, but it is missing this critical midlife window, which coincides with the menopausal transition. So, this is a period of like roughly 45 to 55 years where likely the brain is undergoing these sort of striking changes, but most of cognitive neuroscience had just totally missed the boat. I want to be really clear here. I don't think this was done out of malice. I don't think men were like, let's not study these questions. The point is more subtle, right? It's that you cannot answer questions you don't see. And I think if you're a woman who's lived through menopause, you certainly see menopause. Or if you are a female scientist in the lab, you might say, hey, how come we're only using male animals? So maybe that's the biggest argument for diversity in science, which is just broadening the perspectives and then the nature of the questions that we can actually see and then study.
Missing that midlife window centered around menopause does seem like a big oversight. And fortunately, we have researchers like yourself now addressing that gap. To be charitable to the animal research using mostly males, my guess is that if you're doing like a pharmacological study, that it makes it more complicated when you have to control for different hormone levels. But then again, this is just me thinking out loud, not arguing we should only use males, thinking
Yeah,
that
but stop
even if
there.
it's more complicated, if you're going to give drugs to humans and who go through the same changes, you need to know what the changes are doing. Otherwise, you get this woman's health gap, right?
That seems reasonable, but there's a fatal flaw in your argument. You said, I think that having to control for hormones might make it harder. That's an empirical question, right? So what you're implying, I think, is that females, because they have this estrous cycle, if we're talking about a rodent, they have this 4 to 5 day estrous cycle, they must be more variable, right? That's an assumption that should be tested. And sure enough, when Annalise, this was a paper led by Brian Pendergast that came out just a couple years after their seminal germinal original survey, they asked this question. So they pulled 10,000 different traits from these animal-based studies. I'm talking cell signaling pathways all the way up to behavior. Doesn't matter. Whatever the dependent variable was, they grabbed it for any study in which they had reported sex, that these data were collected in males and females. And what they were interested in is not looking at the mean difference, right? Like males are here, females are here, but the dispersion around the mean, coefficient of variation,
Mm-hmm.
variability. Is there a difference between males and females across 10,000 different traits? What do you think they found?
I guess there's a
I'll
lot
just
less female variability than they expected.
No, it's more than that. It's more than that. There was not a single variable in which the females were more variable than the males. And flipping that, there were several, like a dozen or more variables in which the males were the more variable of the sexes. So if you want to talk to me about getting rid of unwanted, unexplainable variance, get rid of the males because the females are the more stable of the sex. And this was importantly looking at female animals unstaged. So they were allowed to cycle naturally. So even taking into account the estrous cycle, females are less variable than males. So this is a great example of how convention gets passed on in science, and it's a danger, right? Like we have these reasonable assumptions, and I would have guessed the same thing. But unless you empirically test those questions, it can lead to 50 years of science leaving one group behind for no good reason. It's a big problem. And,
Mm-hmm.
there's other arguments that scientists make and grumble about when it comes to sort of inclusion of both sexes. You talk about housing cost of per animal per cage per day. Money's a solvable problem. Um, but I mean, this myth of variability really has to be eradicated. And for anybody listening in this space, I just, especially being there in Cambridge, go look up Becca Shansky because she's published some really important thought pieces on this, really arguing against the myth of this. In fact, she and oh, I'm blanking on her co-author's name, but she just had one in Nature Neuroscience last year, I want to say. Um, Talking about this myth and talking about the fact that, including women's health as this major sort of driver of the kinds of question we ask is going to take a global shift in science culture because
Mm-hmm.
these sorts of myths are so deeply ingrained. Um,
Now that I'm taking a moment to think about this variability hypothesis, it's reminding me of some other evolutionary theory that goes in the other direction that does in fact predict that males tend to have more variability. And maybe that has something to do with like more of a high risk, high reward strategy where like, sperm is relatively cheap, eggs are relatively costly, and there's more investment, especially in mammals where you have to go through pregnancy and all this stuff. So then that could lead you to the opposite conclusion then. Like if we want more robust findings, why not go heavily in the female direction? And this could be related to another assumption that I have that maybe you can slash down. So when I'm thinking about brain development very early, like prenatally, there's this assumption that like the female brain and body is sort of the default human blank slate, and then it's androgens that viralize that, and it starts to take on more male characteristics. That's been sort of a background assumption for me in thinking about the effects of testosterone on early brain development. Do you think that I should work against that?
I think that's a strong and dominant and still in the textbooks theory. So I'm not going to say that it's that testosterone or androgens in general, whether you're talking about Mullerian inhibitory hormone plus androgens, guiding all aspects of sexual differentiation. Those are clearly really important drivers. But I think as the science emerges, we now realize that there's all kinds of factors that are necessary for the sexual differentiation leading to the female direction too, that it's sure in very specific senses, in some cases the mere absence of testosterone will lead to the production of certain secondary sexual characteristics or sex organ production or whatever. But there's also real cases as the science emerges that you need specific factors. I'll withhold judgment until the science catches up a little bit.
This brings up another interesting assumption and probably a gap, a historical gap in the research, which is thinking about the role of estrogens on male development. And I assumed that it just wasn't that significant until I learned about complete androgen insensitivity syndrome, where you can have an XY genetically male fetus and it develops as female, and because the body can't process androgens or testosterone. And not only that, but I assumed that then you would have something like an eternal girl, right? Because you don't have male hormones for male puberty, but nor do you have female hormones for female puberty. But in fact, these kids with CAIS end up going through pretty normal female puberty, except for not having periods because they don't have a uterus, but they do develop breasts and the fat redistribution around the hips and all of that. So then looking into that, I learned basically that males have enough estrogen to go through female puberty, I guess, if not inhibited by testosterone. Is that a right interpretation of that?
Yeah, exactly. Adam, that's exactly it. And CIS is a super interesting phenomenon because it's basically this, kind of genetic quirk where, as you say, their bodies are producing testosterone in abundance, but they do not have functioning androgen receptors, right? You got the lock, you got to have the key. So they don't undergo prototypical, male, they're not shunted towards that sort of like prototypical male form of sexual differentiation, and certainly don't undergo male typical puberty. And as a result, because they're producing these testosterones, all that testosterone ends up getting aromatized into 17 beta estradiol, right? So for those, not in the endo space, these steroid hormones are highly related structurally. So they all originate from this sort of mothership of cholesterol, and then there's certain enzymatic pathways, these biosynthetic pathways that can lead to different hormones. And a lot of what we call hormones are actually pro-hormones. So if you take progesterone can act as in its form as progesterone, but it's also part of the biosynthetic pathway of other hormones that can act in those other forms. Here's the case with testosterone, right? Testosterone can act as testosterone, but it actually almost always acts after conversion into 17 beta estradiol. And that's what you can see on display in cases with CAIS. That testosterone gets aromatized into estradiol, and then suddenly there's all this estradiol to guide development in that sort of female prototypical pathway. And that's what you see. These women with CAAS, they're XY chromosomally, or sorry, genetically male, but they totally identify as female. They, socially, others would identify them as female, and it's only that they lack, yeah, they don't menstruate, and they lack some, internal, those accessory organs. But yeah, that's the power of, I think thinking when you study sexual differentiation in the brain, it really helps you understand the power of these hormones.
Right. So before encountering this, I had a default assumption that I guess you kind of have this symmetry where females have like 10 times less testosterone than males on average. And I guess I assumed that females had like 10 times more estrogen than males. And I guess that is true at peaks of the cycle, like during ovulation, but then at at the more dormant stages, you don't see as much sex difference. Is that right?
Yeah, and even if we think about postmenopause, um, they're about equal. So a postmenopausal woman, the amount of circulating estradiol that she has that's detectable in the serum is about on par with that that you would observed in an age-matched male.
And do you see the same neuromodulation of estrogens in males?
That is a great question. Who fed that to you? Yes. So I mentioned, I can't remember if this is in our pre-talk or in the actual interview, but I think it was in the interview. We talked about
Mhm.
28andMe and this study that's sort of densely sampling a female over time to really understand how these uh, ebbs and flows of hormones can drive changes in the brain, whether it's sort of structure or function. And sure enough, we see these sort of rapid changes coincident with these sort of ebbs and flows of hormones. We, I'm going to admit a bias here. My lab cares a lot about women's brain health. So a lot of our questions are oriented in that way. But we didn't, after the 28andMe paper came out, we were sort of left like, ooh, we don't want to give the wrong impression that females are the only ones that should be thinking about these ebbs and flows of hormones, because that couldn't be further from the truth. And while we know that, like, maybe the public would take that finding and think this contributes to this myth of greater variability or what have you. So we followed 28andMe up with a paper that's just about to be submitted called 28 and He. And this time we took a densely sampled male, so the same sort of precision imaging approach, but we're following a male over every 12 to 24 hours. So AM to PM. So looking at that kind of diurnal cycle, as we know, there's peak production of testosterone and cortisol, and at least in our case, estradiol in the male, and then that production drops anywhere between 40 to 60%, depending on the hormone, in the PM. And so we did this super time lock study where this densely sampled male came in every day, 7 a.m., 7 p.m., and had morning fasting blood draw, MRI scan, and then did it again in the evening. And so that allows us to look at this sort of diurnal variation. And sure enough, the magnitude of change, so we see that testosterone and estradiol, those changes in production are associated with changes in the functional connectome, so measures of coherence across kind of canonical brain networks. And the magnitude of those changes are exactly on par with the magnitude of changes that we saw in the densely sampled woman. The only difference
Wow.
being that, like, in the case of the woman, the accordion was sort of stretched out a little bit because we were looking at changes across, the 30-day or 28-day cycle. And in the male, we were looking really sort of, diurnally. But it just goes to show you that these steroid hormones are neuromodulators for both sexes in a really powerful way.
How about birth control?
Mm, yes. How about birth control? Okay. So birth control is, let's maybe we'll be even more specific, hormonal contraception, right? So hormonal forms of contraception are drugs. This can be patch pill, IUD, um, that are taken for a variety of reasons. In the name is sort of one of them, which is to control conception. I will say that they're used for a lot of other reasons. Endometriosis, right? Controlling the painful periods of that disorder, acne, all kinds of things, even in some cases, migraines. Um, Birth control is an interesting question. Hormonal contraceptives are an interesting question because we, as neuroscientists, know very little about their influence on the brain. Um, and I just was part of a report that came out a couple weeks ago now in Nature Neuroscience. It's a perspective piece that's really kind of trying to um, set the stage for how neuroscience might do more in this space, but also to not call alarm to the fact that even though we're advocating for more research, it's not to say that we should limit our access to these essential forms of medication. So just to be clear,
Mhm.
hormonal contraceptions are a critical medication, a critical for a variety of outcomes, and in their capacity to, kind of blunt hormone production, they're an interesting way of thinking about the role of hormones in the brain. And I think neuroscience could do a lot more to improve our understanding.
In the far future, do you think in the same way, you might do a regular checkup with your doctor, that you would be doing these regular brain scans and seeing how that's connecting to your hormones? And like, do you see some sort of practical implication far down the line where everyone is more informed about like neuroendo?
Sure. And I think it starts with physicians, right? So Johns Hopkins survey that came out a while ago says that 80%, that seems to be the magic number these days, but 80% of medical schools fail to include menopause in the basic curriculum. And for those that do, it's mostly an elective. So if you need the bumper sticker, here it is. Women's bodies are not an elective. We need to educate physicians about women's health, whether it's menopause, menstrual cycle, right? Just neuroendo in general. Um, and then allow that knowledge to sort of trickle down to the general public. We also need more basic science research to inform what matters and what might not. And this is especially true for these questions about the birth control pill or hormonal contraception. Like, there was a study that made waves a while back. This is a Skovland et al. Piece from Denmark linking. So they studied the entire female population of Denmark, which you can do when you have electronic medical records. And they link first use of a hormonal contraception to subsequent increased rate of major depressive disorder, whether that was like ICD-10 codes for depression or filling a prescription for for an antidepressant. That, it's not a perfect study, but there's, offers some suggestion that these sort of major forms of hormonal modulation might impact the brain and thus mood or behavior. But neuroscience hasn't given us, like, lab-based neuroscience studies really haven't given us the tools to understand who's at risk and who's not. And wouldn't it be wonderful if we could say, look, for 90% of women out there, it's totally fine. Like, go forth, young lads and lasses, like, take these things, it's fine. But maybe for 10% of women, be on extra alert. And we could bake this into the medical system too. Like, we could have providers who say, you know what, we're going to have a 3-month check-in, and I'm just going to see, like, are you feeling blue or are you good to go, right? Just have these sort of checks and balances so that we can eliminate suffering, and whilst maintaining our access to these essential forms of medication. But let's understand potential side effects so that they can be mitigated. And maybe I'll just add one more point to that. I think, again, to reiterate, here's a drug that's used for a lot of different reasons. We need it on the market. But for individuals who take it purely for contraceptive purposes, maybe it's a ridiculous way of achieving that outcome. Because, and we are essentially relying on 1960s science. So the pill was released in the United States in the 1960s, and its basic mechanism of action has gone unchanged since then. Why? Okay, here's my final closing thought, because I know we're bumping up against the hour. The beauty of hormones is that they do not have targeted effects. They have all encompassing organizing effects across an animal, whether it's organizational in the sense of, they're sort of literally changing the body over time, or activational, whatever way. Like, hormones are using your circulatory system as their superhighway. They're going everywhere your blood goes. They're getting all of these systems to work in synchrony. They can't have targeted effects. So why would we use endocrine manipulation as the lever to pull for contraception? Because by definition, you might be affecting, the possibility of implantation and controlling reproductive function. But you are almost assuredly affecting all of the other receptors in your body that are responsive to these forms of modulation. I'm more excited by, like, pushing R&D in other directions. So let's think about Polina Lishko, who's working on a drug that alters a single protein in sperm. Doesn't affect the DNA and, the sort of coding DNA, like just a single protein in sperm, to make it swim a little less well. It is as effective as traditional as the pill in preventing contraception, and it has zero side effects.
Wow.
Why not provide that to the market so that women don't have to choose between their brains and their uterus, or between getting pregnant and having severe side effects? In some women, some women, let's acknowledge, have no side effects, and that's great. But, like, why is it even a decision that we have to make when there's other potential pathways that we could invest more R&D in so that these questions become obsolete?
Thank you, Emily, for raising all of these important questions, for slashing back against some unhealthy assumptions some of us might have had, and for your research.
Yeah, Adam, thanks. This was really a lot of fun, and it's so cool to see you bring these different scientific topics and scientists to light. And as a first-year grad student, my gosh, you are just killing it, and it's going to be so fun to watch your trajectory from here on out.
Thank you, Emily.